Welcome, everybody, to the MGH brain-PBM clinic rounds. My name is Paolo Cassano, and today is September 27, 2024. It is my great pleasure to introduce someone that is known by probably all of you, if not, I'm sure most of you. It's Dr. Mike Hamblin, distinguished visiting professor, Laser Research Center at the University of Johannesburg, South Africa. Also editor-in-chief of Photobiomodulation, Photomedicine, and Laser Surgery. Mike has been also instrumental in my path in PBM and Mass General Hospital. So Mike, welcome, and please take it away.
All righty, let me try the screen sharing. So that's working. Everybody can see that?
Yes, we can.
Yeah, jolly good. Well, thank you, Paolo, for the introduction and for finally dragging me to give this talk, partly, I suspect, because I was down in London at PBM 2024, as you can see, and this is an adaptation of one of the talks I gave there. So I'm going to talk a little bit about the history and mechanisms of PBM, and then talk about brain disorders. So everybody wants to see potential conflicts of interest, so there they are. So the history, would you believe we're going back 2 million years, because there was an interesting hypothesis that when humans were evolving from apes, they lost their hair, and at the same time, their brains dramatically expanded. And could these two be linked? So in other words, the less hair you have, the more beneficial light from sunlight you absorb, not just on your head, but your whole body. And this may have something to do with the rapid expansion of the brain. Then moving forward a little bit into traditional ancient medicine, both Egypt, India, and China, Ayurveda in India, TCM in China, that in addition to lots of herbal remedies, they use sunlight. So there was a lot of knowledge about which herbs to take and then how much sunlight to expose yourself to. Moving forward a little bit, von Grotthuss and Draper in the early 19th century formulated the Grotthuss-Draper law, the first law of photochemistry, that light must be absorbed by a chemical substance that we call a chromophore in order for a photochemical reaction to take place. Then, next major figure on the scene, Arnold Rickley who was Swiss, but he was bled in Slovenia, and he got quite a large following for using natural remedies, in particular in the outdoors. And he said, water is good, air is better, but light is best of all. The next figure on the scene is Theobald Adrian Palm, who was a physician in Edinburgh. And he spent some time working in Japan, and he noticed that the area in Japan he was got quite a lot of sunshine, and he noticed that the children there did not have any rickets, which they had in Edinburgh, because Edinburgh was called Auld-Reekie because all the smoke blocked out the sun. So then he came back to Edinburgh and started to use lights on children to avoid rickets. And then, of course, everybody in the PBM field knows Niels Finsen. He won the Nobel Prize in 1903. He only lived for 44 years because he had a hereditary disease, Niemann-Pick disease, and he figured out that the more he exposed himself to sunlight, the better his condition from this hereditary disease. And then he developed the Finsen light, which was a large arc lamp with different filters to get basically blue light and red light, and he treated cutaneous tuberculosis and smallpox. That's why he got the Nobel Prize. So winning a Nobel Prize created quite a stir, and a lot of people got interested in light therapy. Margaret Cleaver was an important one. And this is Kellogg. This is the same Kellogg from Kellogg's cornflakes, interestingly enough. He was a great proponent of natural remedies. And of course, Edison had just invented the electric light bulb. So a lot of people built these electric light baths with incandescent bulbs. And there was a lot of, guides published as to how much light, for what conditions, and what the treatment regimens were. But this really took off, these electric light baths. And in the start of the 20th century, we had this quite popular development of heliotherapy. So people building clinics up in the mountains where people went for cures, and part of the cure was exposing themselves to sunlight. Here's Auguste Rollier, developer of heliotherapy. And you see all these kids out in the sunshine in the mountains, quite cold up there. And Rollier thought the temperature was important. It was clear that these heliotherapy worked better up in the mountains. And the question is, why couldn't you just go on the beach and expose yourself to sun? So he thought it was temperature. My personal opinion is that it's connected to the low oxygen partial pressure up in the mountains, which influences the mitochondrial metabolism. 1916, Einstein proposed the phenomenon of stimulated emission population inversion, but it wasn't until 44 years later that Ted Mayman built the first working laser, which was a flash lamp pump ruby laser. And everybody thinks it must have been some gigantic machine, but it was actually just a little bitty thing. Here it is. So as I say, Ted Mayman built the first working laser. Just 3 years later, Paul McGuff in Boston treated tumors with a ruby laser, and actually published in the New England Journal of Medicine, human malignant tumors. And then over in Hungary, Andre Mester, just 5 years later, developed an interest in laser research. And he got his own ruby laser, attempted to repeat Paul McGuff's anti-tumor experiment. When this failed, he tried to see if laser treatment could cause skin cancer in mice, but it didn't cause any cancer, but he did observe increased hair growth and better wound healing in the mice. And this was really the start of laser therapy. People thought there was something special about lasers. 50 years later, we still think lasers are good, but I don't think there's anything particularly special about them. So that was the history. Moving on to the mechanisms, a very popular and important mechanism involves the mitochondria, which of course are the power plants of every cell, and they take glucose and oxygen, and by a system of electron transfers that go through 4 specific complexes, drive a proton gradient, which produces ATP from ADP. This is called the respiratory chain, and these things are called cytochromes because they're all colored. They're colored because they have heme pigments and other pigments as cofactors, and cytochrome C oxidase complex 4 is one of the most deeply colored of these cytochromes, and that is a very popular candidate as a photoreceptor. So here you see a little cartoon of cytochrome C oxidase, which is unit 4. It's a complex molecule with 13 separate subunits, 2 heme centers, 2 copper centers, each of which has an absorption spectrum, and the oxygen is binding to these heme and copper centers. And the theory is that if nitric oxide can inhibit the binding of the oxygen, it will suppress respiration, and that this nitric oxide could be photodissociated by light because it's only weakly bound. And respiration would resume. It's known you get more oxygen consumed, mitochondrial membrane potential goes up, ATP, cyclic AMP, you get nitric oxide released, a brief burst of reactive oxygen species, and changes in calcium. All these are known and well established. The question is, why do they make a difference? And the reason they make a difference is there are several transcription factors that can be activated by these, we'll say, primary products of photobiomodulation. In other words, the ROS, the nitric oxide, the ATP, and the cyclic AMP. 2 of these transcription factors, one is Jun-Phos, which is AP1, the other is NF-kappa B. The ROS can activate protein kinase D, which degrades I-kappa B, allowing NF-kappa B to go to the nucleus. So between these 2 transcription factors, they will potentially cause the transcription of maybe 200 separate genes. So these are highly pleiotropic transcription factors, and many different proteins will be synthesized if these are activated. And a lot of these proteins are involved in growth factors, extracellular matrix, proliferation, cell motility, and anti-apoptosis and pro-survival proteins. There's many, many more, and you can look them all up, but it does make a big difference. And it may explain why a single exposure to light can have effects that last for hours, days, weeks, or in the case of some animal experiments, even months after a single exposure to light. So they can make a big difference. So this change in mitochondrial metabolism is called oxidative phosphorylation. You tend to switch from glycolysis to OXPHOS. This has 2 consequences. First is stem cells. The stem cells and their hypoxic niche carry out glycolysis, but when their mitochondria are activated, they have to leave their niche in search of oxygen and start to proliferate and differentiate according to the cues they encounter. The second major consequence is to do with inflammation. So the macrophages that have an M1 phenotype are pro-inflammatory. They secrete all the pro-inflammatory cytokines, but they rely on glycolysis for their metabolism. When OXPHOS is activated, they switch to the M2 anti-inflammatory phenotype, and the cytokines change broadly to anti-inflammatory cytokines. And in the brain, the microglia, M2 microglia, can carry out phagocytosis, such as amyloid plaque, for instance. Another photoreceptor is these light heat activated ion channels. So they're TRP, transient receptor potential ion channels, and TRPV1 vanilloid is an important one. That's the receptor for hot chili peppers, and appears to be activated by near-infrared, infrared wavelengths, possibly by a very localized nanothermal effect. But it also can be activated by blue-green light. When it's activated by blue-green light, it's absorbed by opsins. When it's activated by infrared, this is probably due to water producing localized heating. So here's the signaling gated on heat light gated TRP ion channels. They're calcium channels. Blue-green light is absorbed by opsons. Infrared light is absorbed by water. You get a conformational change. The ion channel is open, calcium goes in. These ion channels are present in mitochondria, but the overall result with ROS and ATP is similar to that you get when you activate mitochondria. So it's really just the first stage, which is different, the photoreceptor stage. There's one other possible mechanism is worth talking about, and this is to do with this interfacial water in ATP synthase, which is a molecular rotor, turns around to generate the ATP. And if the viscosity of water is reduced, the speed of the rotor is increased and you can get more ATP. So that's another possible mechanism going via water. So it's been known for quite a long time, it's becoming increasingly appreciated that photobiomodulation does not only have local effects, but has substantial systemic effects. So there has to be some mediator that can be transferred around the body, presumably via the blood circulation. And one candidate for this is these circulating cell-free respiratory competent mitochondria, which were only recently discovered in 2020. And they said, does the blood we thought to know so well contain elements that had been undetectable until now? Which is quite interesting, actually. So over the years, my laboratory at Mass General Hospital in the Wellman Center for Photomedicine did quite a lot of work on traumatic brain injury because we had funding from the US Department of Defense, and TBI then was a really big deal in war fighters with blast injuries from IEDs, and all sorts of things, and there was a lot of funding for TBI. So we had basically worked on mice and rats, and we created TBIs with various models, to direct impact or a controlled cortical injury. And we had various assays, performance assays, learning and memory assays. We had different kinds of light delivery, whether they're lasers or LEDs. And these are some of the papers we published, and I've put in red some of the important findings. That we got increased brain-derived neurotrophic factor, BDNF. We've got increased synaptogenesis and increased neuroprogenitor cells. So synaptogenesis is the formation of new connections between existing neurons. Neuroprogenitor cells are, some people call them neural stem cells. They can go on to form new cortical neurons. And this is, a sort of overview of some of the mechanisms that go on. And there's a lot of different processes here, but you will find that all of these processes, there is documented evidence that they occur. Quite often in mice and rats, because it's difficult to, do invasive assays on human brains, unfortunately, because we'd see all these things. But new blood vessel formation, less swelling, better lymphatic drainage, increased blood flow and oxygenation, anti-apoptosis, a lot of brain conditions, your brain cells start to die, so you can prevent them dying. Antioxidant proteins, anti-inflammatory, and then these synaptogenesis, neurogenesis, and neurotrophins. So all of these have been shown to occur. It's not clear that they all occur for each and every brain condition, but that all of these are generally good for the brain. So what kind of devices do people use? And the answer is it's a whole army of different devices. Some of them are lasers. This was a laser. A lot of them are LEDs. Some of them are on the forehead. Some of them are helmets that go over the whole head. There's some more fancy ones here because as time goes on, the technology gets better and more user-friendly. But basically, they're putting red or near-infrared light onto the head. I just said that there's a systemic effect. So can you put light onto other parts of the body? And the answer is probably yes. But it makes sense to put it on the head if you have a user-friendly helmet and you want to treat the brain. That's the obvious thing to do. So to go through some of the clinical studies we've done over the years, this is Marnie Naser, first 2 case reports of chronic TBI patients. This is the MEDEX cluster. First case, a lady, car crash 7 years before. Her ability to work improved tenfold and got a home use device to use. Another lady with multiple concussions and PTSD. She was given a home unit and she again could return to work. So these, chronic TBI can destroy your life for years and years. And if photobiomodulation can get you back to something approaching normal, it's obviously a big deal. So Marnie was excited by these 2 cases and then did a case series of 11 patients. And you will see that out of these 11, 1, 2, 3 had no major benefits, but the other 8 out of 11 all had significant benefits. This was an early study with French, again at Wellman Center, using a 810 nanometer LED on the forehead. And these were patients with severe depression and anxiety. And they only got a single treatment. So they got a single treatment, and 2 weeks later, their depression and anxiety scores had improved dramatically in every patient, actually. It wore off a bit after another 2 weeks, but remember, this was just a single treatment to the forehead. This was Paolo's early study using a laser, again, 810 nanometer laser to the forehead. And he did 6 treatments, which is probably better than just 1. And you can measure the depression scores over time, and they continue to improve after the treatment was finished. This is the Bi-Light or V-Light neuro, as people say, an array of LEDs on the head, plus a little LED in the nose. And the first study that was done with this was a small clinical trial for Alzheimer's by Anita in Toronto. And the results were surprisingly good. So these were treated for 12 weeks, I think twice a week, something like that. And, this increase in the scores, the ADS cog and the mini mental state, these are the type of scores that you'd use for Alzheimer's. These were dramatic improvements. And when they stopped, the scores deteriorated a bit, as you might expect. But then, it's not shown here, but then the patients were given a home use device and they improved again. So, I don't think people know how long this will continue to show benefits if you have Alzheimer's. I mean, obviously not forever, because you're going to die with Alzheimer's, but it will help your functioning and quality of life for some considerable time, which is a big deal in Alzheimer's. This was Marnie work coming up with this theory of intrinsic brain networks. So there are 3 of these, and Marnie thinks the important one is the default mode network, which are connected in geographically separated parts of the brain. So the idea is you can image the activity of various areas of the brain with fMRI, and the idea is that you don't necessarily want to activate all the default mode network. You want to get it back into balance. That is the thing, because there are different nodes. If they're not operating in balance, this can impact brain function. This is some of the fMRI results, looking at these areas composing the default mode network, specifically the precuneus, which apparently is a functional core. So this is kind of tying together a lot of the things that go wrong in your brain, neuroinflammation, oxidative stress, excitotoxicity, mitochondrial dysfunction, neuronal apoptosis. They lead to these bad things, hippocampal shrinkage, cortical shrinkage is like end state in the brain. And all of these conditions, whether they're neurodegenerative or psychiatric, have been shown to exhibit these type of biological processes and these type of morphological processes. So it all connects together, basically. So the last part I want to talk about is this idea of PBMT can promote neuroplasticity and synaptogenesis, because I think it has big implications, especially for psychiatric disorders. We'll go about autism spectrum, drug addiction, post-stroke rehabilitation, ADHD, PTSD, obsessive compulsive, and I think I've got something on bipolar as well. So the idea is if you can kind of stimulate the pathways in the brain to reorganize themselves in a beneficial manner, that you can do good for all these apparently disparate brain conditions. So autism spectrum disorder is a disorder of neuroplasticity, basically. I mean, I'm sure there are other ingredients that go into it, and genetics and epigenetics, but basically the main thing is it's a disorder of neuroplasticity. So if you can stimulate neuroplasticity, you should help. And it's known that neuroplasticity is much higher in children than in adults, and it appears that autistic children respond amazingly well to photobiomodulation. Adults respond as well, but the children do seem to respond better, and people have been a little wary about treating kiddies because there's always the chance that you do something bad, but putting light on their head is unlikely to be bad, in my opinion. So another disease of neuroplasticity is drug addiction. You can detox and withdrawal is only temporary. The dopamine circuits in the brain reward systems are apparently activated. And imaging studies show that even if you've been free for drugs for at least a month, you have these pathways being activated when drug-related cues are shown. So these folks have these uncontrollable cravings, even when supposedly they're free of drugs. This is just saying that basically these aberrant pathways could be benefited by stimulating neuroplasticity. So Fred Schiffer, the one that did the depression study, his claim to fame was this of two minds, that he can, by using his glasses, he can figure out which side of the brain is important to treat. And it varies between different individuals. A slightly controversial theory, but Fred has been working on it for decades. And he studied drug addicts, again, using his little LED, 810 nanometer LED on the forehead, and looking at cravings, and found quite a big effect with a relatively small sample. So 52% of the treated ones decreased their cravings, but only 17% of the sham. So that was quite a big effect. And he's recently published 2 more papers about PBM for opioid use disorder. But it does seem to work pretty well. And you know, it's been doing it for some years now. Whether it will catch on for drug treatment centers, I don't know, but it's a fairly harmless approach to use, and it's difficult to see why people wouldn't try it. Again, this is money. This was people who had aphasia after a stroke. Now, if you had a stroke, you know, all sorts of things can go wrong. Aphasia is one of the things, but even just motor function, you know, various cognitive aspects, but they can all be improved if you can stimulate neuroplasticity. And I think the reason it worked for aphasia, again, is stimulating neuroplasticity. This was one Lou Bannis did with stroke and aphasia, so he uses a superpulse 904 laser, and that worked. He did one case of several case studies with ADHD, again using the 904 superpulse laser, and that worked. PTSD, I can't trace any human studies, but Zhang, who worked in Louisiana, did animal studies, and it worked quite well for PTSD in rats. He published a couple of papers. So again, you would expect PBM to work for PTSD in humans. This was Fowler's just a proposal, I think, to do it in obsessive compulsive disorder, again, as a classical condition where neuroplasticity should help dramatically. Bipolar disorder, a couple of, this was Paolo's proposal, but there's another paper showing that older patients with bipolar transcranial PBM improves cognition. So it should work in bipolar. New applications, I don't think I've seen any reports yet, but going on the same thing, that any kind of addiction, whether it's alcohol, drugs, nicotine, sex, internet addiction, all these are, you know, popular addictions in the psychiatric community to treat. Eating disorders, anorexia, bulimia, compulsive eating. I mean, you can die of anorexia, quite a lot of people do. And then schizophrenia and personality disorders, I'm not enough of a psychiatrist to know whether you'd expect them to respond well to stimulating neuroplasticity. So this is just like the conclusion. This is kind of segmenting all the different brain disorders that can be treated with PBM This is acute things that happen. You have a stroke, you have a head injury, you have a heart attack, your brain starved of oxygen, and birth trauma. Psychiatric diseases, you can keep carrying on there. I didn't talk about insomnia at all, but PBMT seems to be very good for insomnia. Neurodevelopmental disorders, I talked about that, autism, ADHD. A lot of neurodegenerative, talked about Alzheimer's, Parkinson's is progressing very well. A lot of people in Australia working on Parkinson's and other kinds of dementia. Make a long list there, but they should all be helped by photobiomodulation. So that was my lab a few years ago now, when still was working in Mass General and funding agencies. And thank you all for listening.
Thanks so much, Mike, for a comprehensive overview. Maybe you can stop sharing so we see the different players. And I see already one question in the chat list talking about the use of red and infrared light as ambient light and a kind of supplementing light indoor. I don't know if you had any comment on that.
Yeah, I think there was one, so I didn't show it here, but I talked about it at the meeting where they did a trial during COVID. And what they had was a desk lamp that could be, in addition to giving visible light for working with, could give invisible near-infrared light, and they did it randomly. So they could switch on this near-infrared light invisible randomly, and they found that the people that were exposed to this invisible light while working indoors performed better on various emotional and cognitive scores and so forth. So it's likely that this invisible light could be added to indoor lighting without any bother to anybody. It would cost money, of course, and that now that the energy expense is a big deal for lighting, but aside from the expense of the electricity, you could put invisible infrared light into indoor lighting systems. And it might be especially beneficial in hospitals and care homes and places where folks are confined indoors and they don't get out in the sun.
Got it. So I think Marnie has a question.
Yes, I do have a question. Thank you for that very educational talk. I really enjoyed the history. That was really something. My question has to do with football players. And I wanted to know if you have any ideas about how soon after a human gets a contusion or a concussion or, at least a mild, moderate traumatic brain injury, is it safe or indicated to apply the transcranial photobiomodulation? I know in your animal studies with the mice, you waited 4 hours, but you had anesthetized those mice with their, so when they had the cortical impact on the drop weight, they didn't have pain per se. And, here in America, we have American football, and of course you have soccer around the world and whatnot. And one reason I'm trying to figure out how soon to start it is it seems nobody knows much about photobiomodulation, or if they do in the National Football League, we don't know about it. But they did a study in Italy, and I'm coming to the metabolite N-acetyl aspartate, which is a marker for oxygenation in mitochondria in neurons. So, and you can measure that. We saw that in our football players we treated at the VA hospital. And we can increase that with photobiomodulation or infrared light, red or whatever. And that low N-acetyl aspartate And the soccer players that got traumatic brain injury stayed low for almost 3 or 4 weeks. And then at about 3 weeks to 4 weeks, it starts to come up more dramatically. And that was in the brain. And these are humans. But I'm trying to find out how soon after traumatic brain injury can we start to apply the near infrared light in humans. Yeah.
I would say as early as is reasonably possible. So not just the TBI studies, but a lot of stroke studies from Uri Oron's lab. And they looked at different times after inducing a stroke. And they found the PBM was much better if started early. Now, early is a few hours. Maybe 1 hour is too soon, but 2, 4 hours, 6 hours is much better. If you wait a day, you may get some effect, but it's nowhere near pronounced. Now, these are animal models. So, and also they only got one application, with Oron did. But my guess in humans would be as soon as you can.
Well, another issue is I'm talking traumatic brain injury here in American football players, to be practical. And the issue is, and this would be very rare, we don't want to increase bleeding. So if for some reason there was intracranial bleeding, of course we would not want to apply photobiomodulation, because we're just going to have more bleeding. So the issue is, it would be a practical question of how soon can you get a CT scan, which is going to show bleeding extremely well, if not that expensive these days. And then you could start as long as you have no intracranial hemorrhage or bleeding.
Well, you know, Marnie, people have said that if there's a risk of bleeding, photobiomodulation could increase bleeding. There's no evidence of that.
Okay, it's just based on the idea that it got vasodilation. And
yeah. I mean, not unreasonably, but you know, I don't know whether anybody has done animal experiments specifically to see if photobiomodulation can increase bleeding, but I've never seen them. So people say,
either, that's why I'm asking.
People say this, and it's not clear that there is a risk. I mean, I agree you would probably need to do some animal experiments before you said, oh, I'm going to treat intracranial bleeds with photobiomodulation.
I'm not. Okay. Yeah. I know when I was in China, you know, way back 1985, studying acupuncture on the head to treat paralysis and stroke, it was a thalamic hemorrhage, for example. They waited 2 weeks. And I thought, gee, that's a long time. So I was just wondering, I mean, as long as we know that there's no more bleeding, I would think we could do that reasonably.
You know, I'm sure there must be animal models of intracranial bleeding. So, you know, if some experimental lab wanted to test photobiomodulation on those, I think it would be helpful for the wider community.
But if there's no bleeding, then you think that you could start in an athlete who got concussed or whatever, you know, 2 to 4 hours or 6 hours, certainly within the first 24 hours. And that's what I wanted to know.
The earlier, the better.
I know. I would think that would be my opinion too. So I just don't know how to get the information to the football player. I mean, in Miami, we have this quarterback, this famous quarterback, Tua Tagovailoa, and he got very, very pretty serious. He's had 3 major concussions, I guess, in 3 years. And he's now on injured reserve for 4 weeks, not playing. But I want to know, hello, is anybody applying photobiomodulation to his head in the NFL trainers or physicians? Or it's mostly neurologists that control this, as I understand. They're the doctors they go to. But I don't know if they know anything, and I don't know how to get information to them. I mean, I've tried this before with famous people. I never get anywhere. So I have no idea how to contact the National Football League. Maybe Paolo knows how to.
With the portable battery-driven helmets they have now, you could have them at the side of the pitch. You know, when somebody's carried off on a stretcher, you could get a helmet on their head. Seriously.
Yeah, you could get there's Tom Kerber has really good helmets up there in Toronto, and they can be moved around. That's true. That'd be even easier than having to hold something. You just have to know, I think, they have no bleeding. That should be straightforward. Okay, thanks very much.
It's
There's
a very important question. I know there are some others. I will add that I agree that early intervention might be the best. And at the same time, there's a question of what would be the dose and also the continuation. Unfortunately, the acute TBI study run at Mass General Hospital by the Wellman Center didn't show clinical benefits, although it showed connectivity changes. And also the NEST studies in stroke with acute intervention within 24 hours overall didn't show benefits. And there were reasons, apparently mixed strokes that also were some maybe too in-depth. But certainly something that needs further studying.
Well, I mean, the Nest one was very impressive success. Can't forget that, Paolo. It's just that
right
when they expanded the thing all the way to Nest 3, they just lost control of the whole recruiting situation.
Absolutely. I see questions here. So I see a lot of congratulations. Then there's a question from Liz Corcoran. The theory of concussion is that of microbleeds. So whether there's a concern of microbleeds in concussion. And then we have a comment from Joel Cervantes. If a brain bleed is suspected, I would recommend an MRI over a brain CT scan because after TBI, microbleeds, if present, takes at least 3 weeks to dissipate. We would need to take into account the impact of free blood in CSF and brain tissue. Okay, Paula, sorry, I saw your hand before and then I couldn't see you anymore. Go ahead with your question.
I put it up and down. And first of all, thank you, Dr. Hamblin, for such a great presentation. I was wondering, though, if you could speak to irradiance. Does the power per area of the light make a difference?
Well, obviously, because it governs how much light you get in. The most important parameter is how many total joules you put on the head. And I think Paolo has shown quite convincingly you need 2,000 or 3,000 joules of energy onto the head. How the irradiance governs, how efficient you're going to be in doing it. Depends what device you have. If you have a helmet, then, an irradiance of 20, 30 milliwatts per square centimeter will get you a couple of thousand joules in a reasonable time. If you just have a little focused laser or LED, then the power density irradiance is limited by what heats up the scalp, right? So depending on the wavelength, 500, maybe 700 at the maximum milliwatts per square centimeter, or else it gets too hot and it's painful. The light is, if you have a focus spot from a laser, the light is rapidly diffused in the scalp and the skull. So you don't worry about the focus persisting into the brain. Okay, the light is scattered well before it gets into the brain. So it's just a matter of what is the best way of getting a couple of thousand joules of energy onto the head, basically.
Yeah, Paolo, I've got I'm looking at some of the Photothera data, and they did do a study comparing the effect of PBM applied within 4, 6, or 8 hours, and 4 and 6 hours worked and 8 hours didn't after injury in an animal model. I can't see which animal model it is. I've got some slides and they lack detail from Louise D. DeBoda. But yes, apparently significant effects at 4 to 6 hours afterwards, but not at 8 hours afterwards, using their device, which you're familiar with.
Again, it's a single treatment, so.
Yeah,
The good point, it might well have been, yes, because that's what they did, wasn't it, in those days?
it was a single treatment, yes. No, I think it speaks about the point that Marnie was making. We need to have this probably in the ambulances. so that people get it right away when they have an MBVA or at the side of the football field.
I know about studies that are more involved in pain than brain, but there's multiple post-operative pain studies where they treat immediately post-operatively, within minutes usually, not hours. And the effects are profound, and you only need one. You need just one treatment. And if
And
measured,
also before surgery, if you have that choice.
yes, yes, as well. And certainly we did this at MGH in Charlestown with Mike Whelan, who you probably know, that we compared 1 hour before, 1 hour afterwards, and they got exactly the same results after an experimental TBI on rats, to your point.
Yeah, I mean, when Marcelo did a pain study in mice, he found the maximum effect was 3 hours after the light. So you doing it pre-operatively, depending on how long the surgery is, would appear to be the best thing. I mean, do it afterwards as well, but if you do it pre-operatively, it could coincide with the maximum pain.
Yes. Which then challenges the nitric oxide story. Like, if that's what we're waiting for, is stressed, inhibited mitochondria, and then we put the light in and have photodissociation. But then if you're doing it pre-injury, pre-surgery, that's a completely different state of tissue. And yet
Well, I
we're
mean,
having these profound effects.
He found difference, and he was doing this on normal mice because he was using von Frey filaments, but he found differences in biochemical markers in the dorsal root ganglia.
This is for pain. This is pain threshold, is it?
Yeah.
On
Well,
the feet.
he did actual pain as well, but
Yeah.
But his main thing was pain threshold. But the effect was maximum of 3 hours, and the idea was that there were defined biochemical changes in the dorsal root ganglia, which presumably were temporary, but they were there.
But is this the one where he treated on the foot, over the dorsal, the spine, the neck, and the head?
Yeah.
And did they all produce the same result in the dorsal root ganglion, or just the one over the spine?
It had to be on the nerve pathway between the foot and the brain.
Right. So anywhere on that pathway.
If you did the contralateral foot, it had no effect, and if you did the belly, it had no effect.
Mm. It's continuously baffling. You think you can get a grip of a subject, a bit of it, and you think, I know how this works now, and then you learn something completely new that just kind of puts it all into doubt sometimes, or I don't know.
That's why we have this meeting on Fridays, because then you have the weekend to forget about it and recover. I know we are towards the end here. I don't know if there are any final questions or comments or.
So what do you think about this whole thing of neuroplasticity, Paolo? I tried to sort of paraphrase some of your thinking for you.
Yes. No, neuroplasticity is the holy grail. And if you think about the neuromodulation and the TMS field, all what they talk about is neuroplasticity. How can you change circuitry, connectivity, reestablish connectivity, reestablish balance, as you also mentioned in different circuitry. So I think we have an incredible advantage with PBM because we can't just drive the function. We also have the opportunity to kind of refuel the brain tissue. And the data that are coming also from MGH on TBI, acute TBI treated, are unequivocal that you can affect connectivity and possibly reestablish certain circuitry connectivity that is likely to be healthier. So it's unclear still how that parallels with the clinical effects. And I think we need more studies. So we are submitting, in fact, a DoD grant on TBI, looking at both the clinical effects and connectivity. We're very interested in how much of the effect is driven by entrainment, pacing brain function with lower power LEDs, as opposed to the higher power devices, laser devices that obviously would also have the advantage of re-energizing the brain, as Jacek Domochowski and others have demonstrated with MRS spectroscopy. So it is there. It's our target, understanding that, and will have surely implications for how we define treatment.
Would you expect neuroplasticity to be affected by a systemic effect of PBM? I mean, can you conceive of anything in the blood that could be carried to the brain that would stimulate neuroplasticity?
You know, again, my expectation is yes. And I'm thinking about all the science that is coming from the microbiome and gut, brain-gut axis. And it's clear that there are systemic effects that have an impact on brain pathology and Alzheimer, depression, mood disorders. So, you know, how that could happen in the case of PBM. I mean, you already speculated in many mechanisms, including free-floating mitochondria in the blood flow. I think we need to have experimental designs that really test those hypotheses. And
Yep.
with that, we are at the end, 1 minute past. Thanks a lot. This was a great conversation, great presentation, to be continued. Thank you all. Thanks,
Right.
Mike.
Again.
Bye.
Nice seeing everybody.
Thank you.
Bye now.
Mhm.